All right, so what I'm going to do in this video is I'm going to review photosynthesis, some of the stuff that we went over in class. So the first thing that I want to talk about is the light dependent reactions. And so what we'll discuss here in this whole thing is how the carbon dioxide and the water, along with the light, usually the sunlight, along with the light, gives us glucose and oxygen gas. Okay, and we're not going to worry about how they're balanced or anything like that for right now. But what we're going to do is we're going to talk about how these electrons are going to get peeled off this water molecule. Okay, and we're going to reduce this oxidized form of carbon into a reduced form. So this is the oxidized form of carbon, and this is the reduced, and we're going to do that by adding these electrons onto it with the help of the sunlight. Okay, and as this water loses its electrons, we're going to form this oxygen gas. Okay, so this is a reduced form of oxygen right here, and then this is the oxidized form. So that's essentially what's going on. And sometimes this is referred to as the Z scheme. And so what's going on now is we've got two photosystems, photosystem 2 and photosystem 1. Okay, and they're named, of course, in the order of their discovery. So what's happening then is we've got some sunlight energy that's striking the chlorophyll molecules in this reaction center here. And what they're doing is they're going to transfer the energy into the reaction center of photosystem 2, and they're going to excite the electrons up to a primary electron acceptor. So now I think you would agree that we're missing electrons in here. So what happens is the water is going to donate its electrons. So 2 electrons are going to go into the reaction center to replace that one that was lost. And in that process, that's the splitting of water that's going on. So we're going to get some oxygen gas being generated when we have multiple water molecules getting split. And we're getting some hydrogen ions, okay? And so the same thing is going to happen here with photosystem 1. We're going to get some more light that is exciting an electron up to an electron acceptor as these chlorophyll molecules are relaying that energy into the reaction center. So we've boosted one up. So now the thing is photosystem 2, when this water gets split, this is called the photolysis of water. And this is what replaces those electrons. The electrons that are missing from photosystem 1 are replaced from the electron transport chain, we'll abbreviate ETC, the electron transport chain that links these two things together. So as this moves down the electron transport chain, this electron transport chain is in the thylakoid membrane. And so what we're going to do is this is in the thylakoid membrane. What's going to happen is these hydrogen ions are going to get pumped into the thylakoid space as the electron loses some energy as it moves down the electron transport chain. Small amounts of energy are going to be used to drive these hydrogen ions across this membrane. Now it's a membrane, so remember we've got a nonpolar interior. So these hydrogen ions have to pass through channels during the movement of these electrons down the chain, but also as they pass back through the membrane through this ATP synthase molecule, these things can only, again, pass through a membrane channel because of that hydrophobic interior and their charged ions. So in that process, what happens is we're going to convert ADP plus phosphate into ATP energy. Okay, so this ATP energy is going to be made in the stroma, that liquidy interior of the chloroplast. Okay, and what's unique about photosynthesis too is it's got a second electron transport chain. Okay, so we've got a second electron transport chain there. And what the energy now is going to be doing is as these electrons get transferred down the electron transport chain, we're going to take this NADP plus molecule and we're going to reduce it and give us some NADPH, okay? And this is also made in the stroma, Okay. So these two things get made in the stroma. We have these two electron transport chains, and this whole thing is in the thylakoid membrane. All right. And so what's happening now is these things will be used, the ATP and the NADPH, those are both going to go to the Kelvin-Benson cycle, which we'll talk about shortly, but those are going to be used to help us bring carbon dioxide in so that we can run it through a complex series of reactions to basically give us the C6H12O6, the glucose that photosynthesis produces, okay? But before we look at that, one of the things that I want you guys to see here is these are the photosystems, okay? These are the photosystems. These are the figures from your book. These are the photosystems that we were just drawing and talking about. So here you can see this is photosystem 2. Here's the splitting of water to replace those electrons as they're moving down the chain. They're generating some ATP. And then that other photosystem, photosystem 1, it's going to excite an electron. So the linkage between the two is going to replace the electrons lost in photosystem 1. And then as these electrons move down the electron transport chain, that second one, that's where we're going to get the NADPH. So these two things now are going to go to the Kelvin cycle. So if you look at our figure here, here's your thylakoid. So this is the chloroplast. And in the chloroplast, you've got all of these little thylakoids, these little like poker chips here, these thylakoids. And these thylakoid membranes now, this is the membrane that we were just talking about. So here's photosystem 2. Here's the light striking photosystem 2, excites the electron. Here's where the water is split. So this electron transport chain, which is this yellow line here, you can see that links the two. We're pumping the hydrogen ions into this thylakoid space. Okay, so we've got a high hydrogen ion concentration. So we actually have a low pH in there. Okay, it's acidic. It's becoming acidic. And so as the hydrogen ions move in there, now they're trapped. They can't pass through the hydrophobic interior of the membrane. They can only pass through this ATP synthase molecule. And when they do, that's what gives us the ATP, which goes to the Kelvin cycle. And then that other photosystem, photosystem 1 here, we've got more light striking that, exciting in another electron that's connected to another electron transport chain. And again, this electron transport chain, these electrons are going to be used to reduce this NADP to give us this NADPH, which will also go to the Kelvin cycle, okay? And so I'm showing you this second picture down here. So this one over here, what we have, this is called cyclic electron flow. And so as the electrons basically in photosystem 1, as they start to back up, the second electron transport chain actually runs a little bit slower than this first one. So instead of wasting the energy, what the cell does is it funnels these electrons back into this first electron transport chain to increase the concentration of ATP. So it helps to make a little bit more ATP. While this one is running a little slower, making the NADPH, we can always use more ATP. Okay, so what I described initially is this path here. This would be non-cyclic electron flow. Okay, so that's the non-cyclic flow. And then this is the cyclic flow. So I just wanted to mention that. You can see how it cycles through that first electron transport chain. Okay, so now with the Kelvin cycle. So what I've done here is I've included the figure from your book right here. And this figure over here, we'll call this one figure B. This one is kind of like a hybrid between these two. And this just shows you some of the molecules that you might not be able to see here, say in figure A, okay? But this is the Kelvin-Benson cycle. And these are referred to as the light independent reactions. So these occur in the stroma. Okay, and this is where sugar is made. Okay, so what we're going to do now is in this in the Kelvin-Benson cycle, we have the starting molecule is RUBP, ribulose bisphosphate. And if you remember, we talked about Rubisco. Rubisco is going to add a carbon dioxide molecule to RUBP in the first stage, which is called the fixation stage. Okay, So that's where carbon fixation is going to add this to it. And this Rubisco is a carboxylase. So that's a carboxylase enzyme that's going to add carbon dioxide to RUBP. And what we get is glycerate 3-phosphate. This is the fixation stage. Now in the second stage here, this is the reduction stage. This is where we're going to reduce the molecule. Okay, so this is the reduction phase. So we're going to use some ATP and some NADPH from the light dependent reactions that we just talked about. We're going to use some of that to rearrange the molecule and then reduce it. And we're going to reduce it into a triose phosphate. This is called G3P. So our triose phosphate now is going to get funneled into another pathway to generate the glucose. And so the one of the molecules goes in to make the glucose for every carbon dioxide that comes in. And so the other ones that are left over then go back over to the third stage, which is the regeneration. So in the regeneration stage, we're going to rearrange some triose phosphate into the RUBP using some ATP from the light reactions, so that we can complete the cycle. Because remember, it's a cycle. So what we start with is what we end with. So as this Kelvin-Benson cycle spins, what we're doing is we're fixing the carbon, we're rearranging and reducing it with ATP and NADPH from the light-dependent reactions. We're forming some sugar, and then we're going to regenerate the starting materials, the RUBP, using more ATP from the light reactions to regenerate the RUBP and get it to go again. Okay? So that is the light dependent and light independent reactions. That's a little review for you guys. So if you have any questions, please make sure that you write them down, and I'll see you next time.